So a little bit of picture taking. EEG has been around for almost 90 years now. Here was one of the first labs in the U.S., I think. It was a strange and new idea to record functional brain activity using electrical signatures on the scalp. 20 years later, Gray Walter produced the first image of the electric field on the scalp via analog light bulbs that would flicker when the alpha rhythm appeared and he put them in a topographic array. Of course, no computers. 20 years later, the advent of computers available to psychology labs in particular meant that the ERP technique had responses to stimuli presented in different significance in different contexts began to flourish, and particularly in U.S. labs, has remained the dominant. Applying EEG to cognitive neuroscience questions. 20 years later, then we began playing with MATLAB. Now we had an easy-to-use language, very complete graphics library that we could use to make animations of the field dynamics that are recorded on the scalp. And we could apply signal processing tools easily and share them. And so that was the beginning of the concept of EEG Lab, which became quite widely used. So the prospects for EEG signal processing and understanding and modeling the EEG at the level of the brain source dynamics and not its projection onto the scalp is increasingly good. It requires increasingly more precise measurements, but the payoff will be to actually image brain activity directly, electrical brain activity directly at the scale of and speed of thought and action. So what is the EEG? We know it's only a small portion of brain activity, a small portion of the cortical activity that arrives at the scalp. But which portion and how is it triggered and modulated, and what is the functional significance of all those dynamics that we can see? Now, the talks today have focused on different levels in the hierarchy of spatial scales that define brain activity in a way that none of our technology yet does. In fact, what I like to remember is that at each spatial recording scale, here I'm thinking of electrical recordings, the recorded signal that one gets out of the channel actually is a record of changes in active partial coherence among distributed activities at the next smaller spatial scale. So if you were to record from the bouton of a neuron, you would record the partial coherence in time and space of molecular events, synaptic events. If you record from inside a neuron, you are recording the processes that or data that reflects changes in coherence in space and time of synaptic events across the thousands of synapses of the neuron and extrasynaptic influences as well. If you record from outside the neuron, you're recording the changes in coherence in time and space of the field signals associated with neurons in the neighborhood of your recording. And as you get to larger and larger electrodes, farther and farther electrically away from the cortex itself, you're recording still larger phenomena of spatial coherence that produce the measurable far-field signals that you can record. So brain dynamics are inherently multi-scale. And recently it's been shown again that cross-scale coupling between these scales is not unidirectional, that all implied by the smallest spatial scale, but it's fed back actually so that EEG dynamics or local field dynamics, that potentials that one can record invasively, actually feed back and affect the timing of spikes within neurons and across neurons, and therefore it can affect network activity, it can affect brain plasticity, and so on. So this intercoupling of spatial scales is something that was not in neuroscientific models of the brain activity for a long time, and I think still really hasn't been well incorporated. So the take-home message here is the recorded electrical signal is not actually a unitary signal. It's a summation, a summary of changes, of perhaps diverse changes in neural dynamics or in brain dynamics at the next smaller spatial scale. And of course, we know that the generation of EEG is also extremely, in cortex, is extremely complicated. There's a close interaction between excitatory and inhibitory cell networks within cortex, important contributions of cortical thalamic coupling, sparse but also important connections, cortical connections to distant areas of cortex and to some subcortical areas. What Edelman called the value system modulations associated with different neurotransmitter types, dopamine, serotonin, etc., that immediately affect the dynamics within the cortical column. My mentor Galambos was one of the first to speak openly about neuroglial interactions, which has become a whole field now. And then the ephaptic modulations, the non-synaptic modulations that induced fields or naturally occurring field activities within cortex can affect intracellular dynamics, and in particular spike synchrony. We know too that that there's a layer structure of cortex, and the source and sink layers are not infinitesimally close, but they have important information as well that may not be available on the scalp, although perhaps with very precise measurements one could begin to. Now, the fundamental fact about EEG is that the point spread function is very broad. So on the left, we image synchrony. We imagine synchrony of field activity within a small patch of cortex. And on the right, it has an equivalent dipolar form. The projection to the scalp is equivalent to that of a tiny battery located somewhere toward the middle of it. And this is the projection to the scalp electrodes. So there's only an equator band kind of sites at which there's no projection from this one source, this one effective source, to the scalp. So the scalp sees the sum of many patches in cortex, and when we can actually and therefore any scalp channel, any binary scalp channel, for instance, one site reference to the other site here, may see this source activity or not. Here, this channel would almost not see the source activity since its projection is equivalent at both its cathode and anodal ends. So therefore, this channel wouldn't be sensitive to that source. Now, here's the opposite approach. Given a channel on the scalp, we can imagine this channel, EEG people would call this maybe, what would be F6 to A2 or something. What is it sensitive to? What projections from cortex is it sensitive to? And the answer is all those that are not in bright green here in this map. So in fact, the voltage at time t in this scalp channel represents the dot product between the activity of the entire cortex with this sensitivity map. Red meaning sum the values there, and blue meaning subtract the values there. And that's true for every instant at time t. So although very different things may be going on at these different cortical locations, it's only their sum that you see in a single channel. And then in that sense, the EEG, scalp EEG, is a very fuzzy brain imaging modality, but that is if you try to interpret the scalp signals directly. At the same time, you can see from this that the scalp channel is a spatial filter, sensitive to a particular constellation of brain areas, of cortical areas particularly, and not others. So every channel is a spatial filter. So now the idea becomes, well, how can you optimize the spatial filters to see different sources? And here we bring in the idea of an effective source. What are the effective sources of EEG? And to do this, my colleague Zeynep Kailanachar created a million source cortex. So every mini column scale patch of cortex now was given dipolar projection orthogonal to the local cortical surface. So here, and we simulated 1,000,000 independent EEG-like waveforms at once. And then, but in one, we looked at in one patch, we manipulated the amount of coherence, spatial coherence across the patch. We didn't change the power spectrum or the amplitude of the activity at all. It's always ongoing in every one of these 1,000,000 source voxels here. But we changed the coherence, and we looked at then at the effect on the scalp projections of that change. And this shows the change in coherence. Again, there's no change in amplitude. And so we see that there's almost no EEG projection until there's an emergence of coherence, local coherence. So although nothing has changed about the source signal in terms of amplitude or power spectrum, the effect on the scalp has changed dramatically, which shows that although all the cortex is producing the EEG at all times, actually these, if there are islands of cortical synchrony, then they become very strong effective sources relative to the largely phase-canceled activities at independent activities going on elsewhere in cortex. Walter Freeman first studied these sort of phenomena in spatial temporal dynamics with small grids on the surface of smooth cortex in animals. He saw phenomena he called phase cones, where the random phase portrait across a grid would be replaced by a smoothly outflowing or ingoing pattern. There's some relationship to the somewhat different ideas and phenomena studied by Plentz, as he showed this morning in his talk. Here is a simple particle thalamical model, Stan Anderson at Johns Hopkins. And you see with setting only some cortical-like parameters, you can get these spatially stationary repeating phenomena that Walter Freeman saw. Now here's an interpolation of some ECOG data, much slowed down, of course. And we see, first of all, that the centimeter spacing of ECOG channels is very low pass, spatially low pass filtering. So it's far from the kind of density we would like to have to see the brain dynamics, the cortical dynamics, the spatial temporal cortical dynamics at multiple spatial scales at once. So if one has identified these cortical effective sources, one can, from an MR image, segment the different tissues and build a boundary element or a finite element model of how current flows through the head from the cortex to the scalp. And this is Zeynep's NFT package for EEG Lab that makes such things. And then given some electrical data, EEG, MEG, or intracranial data, as a matter of fact, and an adequate model of the different tissues in the head and their electrical properties, then through signal processing of your recorded data, if you can you can propose an inverse problem. That is, you can show a scalp map and wonder where those potentials in the map come from. And if it is a simple map representing the projection of one effective source, that is one locally coherent activity across a patch of cortex, then it is in principle possible to estimate the location and distribution of that source activity. But how to find such sources, source projections? Well, here we turn to the topic of blind EEG source separation by independent component analysis, or ICA, which I first saw in a paper by Tony Bell in 20 years ago or so. And the concept is this: you're at a cocktail party, you have multiple sound sources and multiple microphones recording the cocktail party noise. And then some French engineers 25 years ago or so began to think, well, can we isolate the individual sound sources in these mixed signals, simply without trying to fit a saxophone to the noise and then subtracting the saxophone and fitting a man's vocal tract, and then which had been the kind of way that the engineers had gone about such problems before. But can we solve this problem by just relying on the fact that the man's voice gives different information, contributes different information to the microphone mixtures than the saxophone? So the saxophone and the man's voice in this ideal situation provide the only two sources of information to the mixtures recorded by the microphone. And by, therefore, linearly unmixing, this is Tony Bell, who worked out the beautiful algorithm for IC that we use. By unmixing the signals, one can only return to the in the direction of more and more distinct information in each signal. One can only arrive at the original information sources in the data. So I had a dim idea that this could be applied to EEG, and because you have the situation where you have propagation to electrodes, linear summation there, and therefore it's possible, I reasoned, that unmixing these situations could arrive at the time courses of activity in independent signal sources, effective sources of EEG that had independent time courses, or maximally. And from that, you could also get their projections to the scalp electrodes, separated from each other. And a week later, we had a NIPS paper whose conclusions, I'm happy to say, are I still have held up over the 20 years, although, of course, we've refined a lot our understanding and methods. So what can you do if you take a high-density EEG data set and separate it out into maximally independent components, maximally distinct sources of information in the data based on their different time courses? Well, you can find eye blinks and their projection to the scalp. You can find lateral eye movements and their very strong projections to the scalp as well. You can find individual muscle signatures in the data and see its spectrum on the bottom here, and the changes in muscle tension across an experiment here. The subject probably was not aware of that, but they're very distinct. And you can find the presence of the heart rhythm in the data channels. You see everything non-green is non-zero, so this is the projection of heart activity into the EEG of this subject. Now, once you can also ignore all those sources and just focus on sources that are completely compatible with sources in the cortex. And here, for one data set, are a set of those. You recognize those by having a signature of that the scalp projection almost completely resembles an equivalent dipole located in the brain. So here, with only 2% residual variance across 256 channels, you have a equivalent dipole model of the source, and that's equivalent to the actual patch of cortex that constitutes the effective source. Sometimes, of course, you have bilaterally coupled areas, and the scalp map reflects this dual dipolar structure. Well, by separating out all the non-brain and brain activities from each source activity, of course, you get much higher signal-to-noise for each of these sources. So you can look at the single trial level at changes in EEG signal at the source level, at the effective source level, without depending on context and so on and so forth. So it makes EEG not fuzzy data anymore, but it separates it into effective sources that contain a great deal of information about the brain dynamics, that underlying cognition. Now, if you looked at EEG signatures, if you looked at experimental psychology papers for decades, you would think that natural cognition involved seeing things, because the visual system was the source of almost all the experiments, and many of these experiments, almost all of these experiments, involved rapid, instant tachistoscopic presentation of some visual stimulus. But of course, we know that natural cognition actually it represents evolution's response to demands of increasingly complex environment in which we need to survive and thrive. And those include, crucially, action, body action, to optimize its results, and social interactions. My point I want to make here is that EEG, and particularly source resolved EEG, can now be used to learn a great deal more about brain dynamics, how our macroscopic brain dynamics, our distributed brain dynamics, and in particular, of course, the cortical dynamics that we mainly see with EEG can help us understand how the brain supports our experience and our behavior. To do that, of course, it's really important not to record just from the brain as if it were in a glass jar. The brain is evolved to coordinate our actions and our interactions. So it's natural to think of multimodal brain-body imaging as a way of the future, and I don't have time to go much. I've thought a lot about, well, what is cognition involved in, other than visual perception or in general sensory perception? And I came across a quotation by Avatar Meher Baba that at least gave me, he said that the three aspects of human consciousness are knowing, feeling, and willing. And my interpretation of that, he didn't give an interpretation in that passage. If you, by knowing, you include perception, but also memory, belief, anticipation, all the kind of cool cognition that has been a topic of much of cognitive science. Then that leaves the feeling cognition. I experience as feeling, which has been shown and understood now to be increasingly to be a crucial part of our cognition that's easy to overlook. That was easy to overlook in the scientific context, but it's certainly you can understand our natural cognition, our behavior, without understanding the deep interconnections between our feeling cognition and our knowing. And finally, there's the willing cognition. What do I intend to do? What am I doing? What effects what I actually do have on my environment, on others I'm interacting with, and so on. So knowing and feeling and willing, I think, is covers the full range of our normal human cognition. So I show a few applications here very briefly in these three areas of source-resolved EEG. So here's a simple faces and houses flashed photo experiment, ERP experiment by Henson and Wakeman. The data is available. And here is the averaged ERP to presentations of faces, so pictures of faces here at time 0. And this is the ERP across all the channels they're recording from. And this particular feature is called in the literature the face response at N170, about 170 milliseconds. So, and it has a scalp distribution that looks like this. It's been much studied in the literature. Well, what does ICA decomposition say about this? It says, well, there are two main sources of this potential. One is on the left and one is on the right, or more particularly, one is predominantly on the right and the other is predominantly on the left. And if you actually find the equivalent dipoles for them, they are in fact in the face area in bilateral inferior temporal cortex, one dominant on the left and one dominant on the right. And that they couple together or they jointly produce most of the N170 feature. Although you see their latencies are not quite the same. So now you can understand at the source level, you can study small details that are buzzed over in the broad scalp data. If you look at the decomposition of the entire ERP bundle of channel ERPs, then you see that something very interesting happens here. The negativity that arises at the scalp, in this case, actually just arrives almost exclusively from these two processes. But if you look at the positivity, the P170, one could call it, that occurs at the same time, you see it's very spatial, temporally complex. But many sources happen at that point to project positivity to the scalp in highly overlapping fashions. So this hasn't been a very popular area, of course, for people studying scalp ERPs because it's too complicated for them to interpret. But now with source-resolved methods, we can actually begin to look at the influence of, for instance, pathological states or different states of consciousness or different individual differences on these constituents of these responses. And in fact, we've had very good success in showing that there's a lot of information in the source-resolved ERP measures that are not available in the scalp data directly, for instance, in schizophrenia. Now, what about feeling? Can you study feeling directly with EEG? And here I point to my colleague Julie Anton. 10 years ago, we did an experiment in which we asked people to imagine different situations in which they would feel different emotions, and we asked them to pay attention to that in their body. Can you feel the anger welling up? Can you feel the joy? And so on. When you feel it, press a button and then hold it as long as you can by staying in that imagination. And then we gave them relaxation instructions between emotions. And there's a paper on this with a kind of unemotional title, but it's really quite a wonderful study, I believe, and about which we're learning still more now. We studied 15 different emotion words, and so they imagined situations in which they would feel all these different emotions. Then we, through a process of spatial separation using IC decomposition, and then frequency domain separation using, again, ICA applied to the log power spectra. And now focusing on just high frequency activity in the brain processes separate from the scalp muscle processes. We found that the state space of the different emotion of the dynamics in the cortex and the different emotions looked like this when projected to 2 dimensions. And you see that similar emotions are clumped together here. And when you look at which emotions people, or a different set of subjects reported as positive versus negative, you see that regression line occurs in this space, in this direction. And in fact, the correlation between the position on this line in this space, found blindly just from the EEG dynamics, and the actual ratings of these different emotions for valence, for being good or bad emotions, the correlation was 0.96. So that in the high frequency activity, also in some of the lower frequency bands, there's detailed information available about the emotion the subject is experiencing. And we found 2 spots in the brain then in which high frequency activity would indicate, commonly across subjects, would indicate the valence of the emotion they were experiencing. Not just good or bad or good or neutral, but in very detailed form with that correlation of R.96. And so, of course, I looked in the functional imaging literature for any parallel to this. At that time, I didn't find anything, but since then, I found a study of good and bad and neutral music, and good and bad actors saying the same sentences with good or bad intent and prosody. And these same areas appear in temporal lobe. So this is a case where source-resolved EEG really first found very precise areas in the temporal cortex in which activity in high-frequency EEG, which should be correlated with. So currently we're looking at this, and we're data again, and using a non-stationarity detection method, we find that this method can actually detect changes in brain state for each of the emotions. And now I'll just skip to my final slide. And that is to say that by studying all these aspects of truly natural human cognition, knowing, feeling, and willing, one can study not just the simplest form of human experience. The applications of EEG to cognitive neuroscience will continue to expand, and they'll become richer with the new signal processing